Hi Nat5s, so here is your final presentation on variation and inheritance, and this is applying it, which is probably the trickiest bit of the content. Okay, so we're looking at genetic crosses, and genetic crosses are useful for lots of different things. So first off, we could look at the offspring, and from the offspring you can then work out what alleles the parents must have had. So you can work back the way. So because you're working back the way, it is called a test cross or a back cross. Okay. You also could work out which allele is dominant and which is recessive, pretty much just by saying which one shows up more often, and the one that does, you go, well, that's the one that's dominant. You can also use crosses to work out how likely you are to get a particular phenotype, and that's quite useful and one that we're going to use quite a lot. So to do this, to bring in yet another term, sorry, but we need to have them, we will use parents that are often you'll be described as true breeding. Now true breeding just means that they always pass on the same thing. So if they always pass on the same thing, they're breeding true, then we now know that must mean that they have the same alleles because they're never passing on anything different. So if they're passing on the same alleles, then they must be homozygous. So that means that we're looking at either a big A, or whatever letter you've used, or little a. Okay, in this case, if we had true breeding parents, then that tells you straight off the purple must have been dominant. So we'd give the purple the big colour, the big letter, but P is not the greatest letter, so let's go for F for flower. So the purple would be given big F, and that means that purple one must have been big F, because I'm saying if they were true breeding, which means this one must be little f, and that one must have had a big F and a little f, because he would have got a little f from there. So you can do quite a lot just by looking at what you've got. Okay, so when we're using genotypes and crosses, I've already said all this in the definitions that you need to know, but just to be absolutely clear, we're going to use a letter for each allele. We're going to use a capital letter for the dominant, small letter for the recessive. We're going to be very careful about which letters we pick so that they look different in their capital and small. We're going to use P for parental generation and the F1, F2 for the first and second filial. Okay, so let's go with this one. We have a pink mouse and we have a white mouse. Okay, so our pink mouse, we're going for a homozygous parental, so big B, and here's our white mouse, and our genotype here is little b, homozygous recessive. Okay, right, we're going to cross these. So we're going to cross these using a particular form called Punnett squares. So here's our parents back in again with their phenotype and their genotype. You basically try to write down everything you can about each one. Right, so then our possible gametes. So what could they actually give? Well, the pink mouse could only give you a big B or a big B. There is no other option. And the white mouse could only give you a little b or a little b. No other option. So this one, not entirely sure you'd have to write it out as a Punnett square, but just so you've seen them several times. Here's a Punnett square. So a Punnett square means that you fill in one side with one parent and the other side with the other parent. Okay, and then we just cross them together. So in this case, our big B from here, little b from here, big B across, little b down, and I'm sure I said before, the standard is always to put the big B before the little b. Okay, so that means every single one of these, they all came out pink. Okay, so our phenotype is all pink, and our genotypic ratio is also all big B, little b. Okay, so that's the F1. If we kept going, here is our F2. Okay, so our F1, taking two of the individuals from the F1, so big B, little b. Now this time the gametes are big B or little b. Put them into a Punnett square, same deal. So bring these ones down and these ones down and fill them all in. And so we've got our big B from here, little b across, little b, and our big B and big B, and our big B and our little b. So this is our possible solutions, as it were, for the F2. So phenotypic ratio this time is going to be 3 pink, because we've got one big B here, and two big B little b's, and one white. These ones have the same phenotype but different genotype. That is quite commonly a question, so make sure you're happy with it. Okay, so this is our F2. You're never going to be asked to go further than that, but this is a really useful one because it tells you if you ever see a 3:1, then you know straight off, well, it's these guys. I'm crossing two heterozygous. So the thing is that 3:1 doesn't often happen. Like it's great, but it's not always the case. So here is Mendel, who figured out a lot of the crosses stuff. This was some of his actual data results. So he was looking at pea plants. So he started off with purple and white. F1 is all purple. That tells you purple must have been your dominant here. And then did the F2 and got 705 purple and 224 white. Now that, when you cancel it down, doesn't even give you a perfect whole number, 3.15 to 1. So that's not 3:1, but it's close. Same here, 2.82 to 1, 3.01 to 1. These are close to 3:1 because it's never going to be absolutely bang on unless you have a massive, and I mean huge, sample. You'll notice this one is getting closer, but look at the numbers that were involved. There were, over 8,000 seeds that they looked at, or plants that they looked at. So the reasons for this, and you can be asked for a reason. This is the one that will always get you the mark. Okay, you don't know when you do the Punnett square and you've got your little cross all lined up here, and you've got, see, that's a big A and a little a and a big A and a little a. You don't know. This big A, it's a 50-50 chance as to which one of these it then joins up with. This one, 50-50 chance here. So you don't know which one you're going to get. It is random. So if the sample size isn't big enough, you might end up with lots of big A's, but not actually enough to tell you anything. Quite often they don't make it, so you might not develop, they can't be counted, and they might fail to germinate if they're seeds, or they might die before you can actually count them. So if you have a plant that didn't make it, that's exactly the same thing as not being able to count what its real thing was in terms of phenotype. Okay, so just to recap, find your genes, decide on the letter, capital and small, give the parent, give your phenotypes and genotypes, put down the gametes, draw the Punnett square, write out each possible genotype and the number of each of them from the Punnett square as a ratio, convert it to phenotypic. So for example, we've got our round and wrinkled in peas. That's a round by round, a big R crossed with a big R little r. So there's your possible gametes. Here is our Punnett square. F1 is 2 big R to 2 big R little r. That's our genotypic ratio of 1, and it's a 1:1. But our phenotypic is going to be all round. Okay, that's everything in this one. You probably want to do quite a bit of practice on these crosses.